Hi students, welcome to the second part of the chapter on the structure and function of DNA. So ribosomes are the organelles that actually make polypeptides, and remember that polypeptides are the building blocks of functional proteins. Ribosomes themselves are made up of two protein subunits, in blue here, and they contain ribosomal RNA or rRNA. So there are actually three different types of RNA. There's the rRNA, mRNA, which is messenger RNA, and there's transfer RNA. So mRNA contains that specific sequence of nitrogenous bases, and remember that three of these nitrogenous bases specify for one amino acid. So it basically tells the transfer RNA which amino acid it needs to add to the growing polypeptide chain. And of course, this is a very simplified version of this process, but for the purposes of this introductory class, basically the information on the slides is what I need you all to know. So as a recap, DNA is the molecular basis for the instructions for all living organisms. But in order to get a fully functioning organism from that blueprint, we need RNA as the middleman for the transcription process, and then we also need proteins to be translated from that RNA molecule. So DNA replicates itself, it makes copies of itself. RNA is transcribed from a DNA template molecule, and proteins are translated from that RNA molecule. So when DNA is transcribed, the result is an RNA molecule, and RNA is then translated into a sequence of amino acids in a polypeptide. Transcription occurs in the nucleus, and translation occurs in the cytoplasm. So again, we have a depiction of a cell here. This purple circle is the nucleus of that cell that contains the entire organism's genome, so all of the DNA is within this nucleus. We have transcription occurring here with the help of the helper enzyme RNA polymerase. We have a template DNA strand being used in the process of transcription to make an RNA molecule. Outside of the nucleus, we have the process of translation, in which case the RNA is actually translated into a protein. Your book has a little bit more detail about this process, but again, for the purposes of the homework, your quiz, and the final exam, basically what I tell you in the lectures is the important information for you to know. A mutation is any change in the nucleotide sequence of DNA. So, for example, we have a normal hemoglobin DNA section here, and this particular codon specifies cytosine, thymine, thymine. Sometimes mutations occur within DNA molecules. In this particular case, we have an adenine substituting for a thymine. So what happens is, during the transcription process, the wrong messenger RNA molecule is made. So instead of being GAA, it's GUA. Since that T was substituted for an A, the complementary nitrogenous base is going to be a U instead of an A, and that actually changes what amino acid is made. So that actually affects what protein is made and thus its functionality. So for a normal hemoglobin, we have a particular amino acid here, specified by the first 3 letters of its name, GLU. And for this mutated sickle cell hemoglobin, we have an entirely different amino acid made. And the amino acids are specified by just the first 3 letters of their full name. So in this case, it's VAL. This is a different amino acid, and it's not going to function the same as the normal hemoglobin. Although mutations are often harmful, they are the source of the rich diversity of genes in the living world. So really, mutations contribute to the process of evolution by natural selection, but it's important to note that mutations are not the only driver of natural selection. There are many other drivers of evolution, including genetic drift, such as the founder's effect, which we'll get into later when we talk more about evolution. There's natural selection itself, which really acts on existing mutations and populations to suit them to their environment better. So mutations are important, but they are not ultimately responsible for all of the diversity that we see. They're just a major building block of that diversity. Okay, switching gears a little bit here, let's just talk briefly about viruses and other non-cellular infectious agents. So viruses are simply DNA or genes wrapped in a protein coat. So this is a depiction of a virus here. We have DNA in the middle, and it's surrounded by various types of protein. Viruses are not considered to be alive, so they do not have all of those 7 properties of life that I talked about during the first chapter lecture. So they are not composed of cells, and they cannot reproduce on their own. And as all of us know, unfortunately, viruses sometimes take over our own bodily cells and cause us to be ill. They are basically using us, using our own cellular machinery to reproduce. Bacteriophages are also known as just phages, and this literally means bacteria eaters. These are viruses that specifically attack bacterial cells. Once they infect a bacterium, most phages enter a lytic cycle, which is a type of reproductive cycle. And some phages can also reproduce via a lysogenic cycle. Phages are kind of scary looking. They look a little bit like tiny aliens, at least what we often popularly depict aliens as looking like. And they are able to attach to bacterial cells and insert their own DNA. So these are the lytic versus lysogenic cycle, and depending on what type of virus it is, it may undergo one or both of these cycles. So here's a phage, again a virus that infects a bacterial cell. The first stage here, regardless of which cycle it's going to go through, is it has to attach to a host cell. So here we have a bacterial cell, a very simple cell that contains its own DNA in the form of chromosomes. So that phage is going to insert its own DNA into that bacterial cell. So the second part of this cycle is the phage DNA circularizes, which means it basically becomes ring-like. So if it enters the lytic cycle, the third stage is going to be the new phage DNA is basically synthesized within the cell. So the phage uses the bacterial host cell's own organelles to make its DNA. So the DNA is synthesized within that bacterial cell, and then the proteins can be made from that DNA. And then within that bacterial cell, complete phages assemble, so it has basically successfully reproduced here. And then the host bacterial cell lyses or bursts, and it releases these phages. So what has happened here is the phage has taken over the cellular machinery within that bacterial cell to reproduce its own kind and further infect more cells. With the lysogenic cycle, after that phage DNA circularizes, it could enter into this prophage stage, in which the phage DNA is inserted into the bacterial chromosome. So it basically becomes part of the bacterial chromosome. Then the prophage is replicated at each normal cell division. So it basically disguises itself as part of the normal genome within that bacterial cell. So cell divisions occur, and it's incorporated within that bacterial cell's genome. So that is the alternate cycle called the lysogenic cycle. Most plant viruses use RNA rather than DNA as their genetic material. And examples of these are the tobacco mosaic virus and the leaf curl virus. And remember, RNA is also a nucleic acid. It's ribonucleic acid rather than deoxyribonucleic acid. So the main difference between RNA and DNA is just the type of sugar that it uses. So it's ribose instead of deoxyribose. Many animal viruses have a membranous envelope made of phospholipids that allow them to enter and exit cells. So they can basically easily slip into and out of host cells. Examples of animal viruses: influenza, commonly known as the flu. Of course, there are many different strains of the flu. Herpes simplex, HIV, which leads to AIDS, and mumps. And this is an example of a virus entering a host cell. We have a protein spike here, a protein coat, and an envelope. And within this is the viral RNA genome. This virus enters a host cell. The proteins basically separate from that RNA genome. So we have a free-floating RNA virus here. And then the RNA synthesizes by way of viral enzymes. So there's enzymes that come with this, and those allow the virus to take over the host cell's machinery. So protein synthesis can occur. All of the proteins that that virus needs to function can be translated from the RNA, and that produces new viral proteins. And the RNA can also synthesize new viral genomes. And if you put both of those together, you have a complete new virus here, and then it can exit the cell and probably infect other cells within that host if it's a multicellular organism. Human immunodeficiency virus can lead to acquired immunodeficiency syndrome, which is AIDS. It's an RNA virus, and it's what's known as a retrovirus. So it reproduces by means of a DNA molecule, and it reverses the usual DNA to RNA flow of genetic information. So it goes from DNA to RNA. Instead of that normal flow of information, it goes backwards, RNA to DNA. And it carries molecules of an enzyme called reverse transcriptase, which catalyzes the synthesis of DNA on a RNA template. So that reverse transcriptase is an enzyme which allows that flow of information to be reversed. I'm going to go ahead and pause here and finish up the next couple of slides in the next recording. Thank you for listening.